Hello, my name is Sahel Shah. I'm a fourth year medical student at King's College, and this is my critical appraisal. The paper I've chosen to present on today was published in the New England Journal of Medicine earlier this month. It's a study on the safety and efficacy of the mRNA 1273 vaccine designed by Moderna against the coronavirus disease caused by SARS-CoV-2. As we all know, the coronavirus disease was declared a pandemic by the WHO in March last year. A safe and effective vaccine will be at the heart of any attempt to contain the pandemic and curb the devastating medical, economic, and social consequences of the disease. The Moderna vaccine is the third such vaccine to be approved for use in the UK, and 17 million doses have been ordered by the UK government. This is a substantial investment and constitutes the biggest vaccination effort ever seen in the UK, with a planned 2 million vaccinations per week at over 2,700 vaccination sites across the UK. But how can we be sure that the decision is evidence-based? The paper we are discussing today is the only study referred to by the MHRA, the Medicine and Healthcare Regulatory Agency, in their decision to approve the vaccine in the UK. If we were to base national policy on a single paper, one would hope that the paper has something to it. To appraise the paper, I will be using a CASP checklist tool that provides a framework for a systematic appraisal. On this slide, you can see a full breakdown of the questions we will be asking. The first question to address is if the trial considered a clearly focused issue. For this study, the intervention is very specific. It's the mRNA 1273 vaccine. The control to which the vaccine is to be compared is also clear, a placebo of saline solution. The study population of a little over 30,000 was recruited across the US and consisted of individuals over the age of 18 who had no known history of SARS-CoV-2 infection. The endpoints on which the intervention is to be assessed are also well defined. The efficacy endpoint is the prevention of symptomatic COVID starting 14 days after the second dose of the vaccine. The safety endpoint is the occurrence of solicited adverse reactions in the 7 days following each dose and unsolicited adverse reactions from the first dose to a period of 28 days after the second dose. It should be noted that the trial is still ongoing. This paper is based on a final analysis that was conducted when the number of COVID cases reached 151, as this figure was calculated to provide statistical power in evaluating the vaccine. The study will continue to follow up on the efficacy and incidence of adverse events with patients for a period of 2 years and publish an end-of-study report. All in all, the main issues to be addressed are clear and well defined. The next question is whether the assignment of patients to treatment was randomised and if all patients were accounted for at the end of the trial. The participants were randomised in a blinded manner using a centralised interactive response technology, fancy way of saying computer. A 1-to-1 ratio of vaccine to placebo ensured similar numbers of participants in the two arms of the study. The study population was stratified by age and risk to ensure that 3 subgroups of interest were adequately represented for meaningful analysis and to avoid confounding. In this slide, you can see a full breakdown of the outcomes for all the participants that were part of the randomization process. There were a handful of deviations from protocol, and they are clearly documented and accounted for. There were a number of reasons for the deviations, and these participants were excluded from the per-protocol analysis. The 2 main deviations were a positive test for SARS-CoV-2 on the day of the first injection, and participants who discontinued without receiving a second dose. A handful of patients received the wrong injections and were also excluded from the per-protocol analysis. Although the patients mentioned were not included in the per-protocol analysis for efficacy, they were included in the safety analysis as they had received the first dose of the vaccine and were followed up with safety in mind. The study followed the protocol for an observer-blind study. The investigator, study staff, study participants, site monitors, and sponsor personnel were blinded to the administration of the vaccine until the study end. However, there were some exceptions, some of which were understandable. For example, unblinded personnel were used to prepare the vaccine for all participants. These persons were accountable for vaccine management, documentation, accountability, and preparation. The injections were prepared in a secure location, not accessible to blinded staff, and an opaque sleeve was used to cover the syringes, as the placebo and vaccine had differing appearances. Similarly understandable was that designated team members at Moderna had unblinded access to the data to facilitate interface with the regulatory agencies and the Data and Safety Monitoring Board. However, there was some unblinding that seems unnecessary. Unblinded personnel were used to administer the vaccine to the participants. This should have been avoided to minimize bias. It opens up the potential for interactions that may reveal to the patient the nature of the intervention they are receiving, perhaps through a slip of the tongue or subtle communications in language and demeanor. Similarly, the statistics and programming team was unblinded to the data when conducting their analysis. Again, this should have been avoided if possible. It opens up the possibility of bias in favor of using statistical measures which support a desirable analysis. On the whole, the study uses a good protocol for blinding. However, as discussed, there are certain vulnerable areas that could have led to bias. Next, we consider if the two arms of the trial were similar in nature at the start of the trial. In the supplementary appendix of the paper, there is a comprehensive breakdown of the characteristics of the participants randomly assigned to each arm of the trial. Both groups are remarkably similar with respect to age, gender, ethnicity, BMI, and other notable risk factors for COVID, such as chronic lung conditions, diabetes, liver disease, and HIV. Now we consider if, aside from the intervention, the two arms of the trial were treated equally. The setup of the trial was such that patients were trained to use an e-diary app to monitor progress following intervention. Daily and weekly updates from the patients formed the basis of the dataset. The training the patients received was done on the day of the initial dose and was uniform across the groups. For safety analysis, the diary was to be filled on the day of each dosing and daily for the following 6 days. Participants were prompted to answer on local adverse reactions such as pain at injection site, erythema, swelling, localized axillary swelling, and systemic adverse reactions such as headache, fatigue, myalgia, arthralgia, nausea and vomiting, body temperature, and chills. Again, both groups used the same app and the same prompts. To monitor the occurrence of COVID symptoms, the e-diary was used with check-in frequency of once a week. The patients were trained to contact staff immediately if they experienced symptoms of COVID or came into direct contact with a confirmed case, and information on how to do so was given to both groups. If information in the e-diary identified a relevant safety event or COVID symptoms, a follow-up call by the study team was triggered and the patient called in if needed. The protocol for the groups showed no difference, and it's fair to say both groups were treated equally. We now come to the crux of the matter and consider the treatment effect. The primary efficacy endpoint was the efficacy of the mRNA 1273 vaccine in preventing a first occurrence of symptomatic COVID, with onset at least 14 days after the second injection among participants who were seronegative at baseline. The intention of the authors was to conduct a primary analysis when there were 151 cases of COVID in the study population, as this figure would provide statistical power to test the efficacy of the vaccine. However, this figure was surpassed quicker than expected, and the primary analysis was completed when 196 cases of COVID-19 were diagnosed. Of the 196 cases, 11 were in the vaccine group and 185 in the placebo, indicating 94.1% efficacy of the vaccine as compared to placebo. This, of course, is an extremely positive outcome in favour of efficacy. The 95% interval for the efficacy was 89.3% to 96.8%. One of the more interesting secondary endpoints was the efficacy of the vaccine in the prevention of severe COVID, defined as COVID additionally satisfying one of the criteria on screen. 30 cases of severe COVID manifested throughout the study population. All 30 cases were in the placebo group, suggesting a vaccine efficacy against severe COVID of 100%. The data on severe COVID is not complete enough to assert this conclusively, but the result is promising nonetheless. With regard to safety, the final paper presents data which points out an increase in solicited adverse events in the 7-day period following both injections. This was true for events local to the injection site and systemic events. Much of this effect will be accounted for by reactogenicity and is to be expected following a vaccination. However, the published paper fails to strike the right tone in its discussion of the safety data, and there are certain aspects of data that have been overlooked and are worth attention. The data regarding solicited local and adverse effects reported in the patient e-diaries shows considerably more events in the vaccine group compared to placebo. This is clearly presented in the paper's analysis. However, neglected in the final paper was a clear breakdown of the grading of these events and the implications. The grading system can be found in Moderna's protocol document. Grade 2 was defined as the need for repeated use of over-the-counter pain relief. Grade 3 was defined as preventing daily activity or the need for a prescription pain reliever, the emphasis being on prescription, which implies a visit to a healthcare provider. And Grade 4 was a visit to the emergency department or hospitalization. Data in the supplementary appendix shows that 59% of the vaccine group, who were screened to be healthy at baseline, suffered a grade 2, 3 or 4 event following the second dose. The most common events were injection site pain, headache, fatigue and chills, with a mean duration of symptoms of 3 days. Notably, 19.6% of the group suffered grade 3 or 4 symptoms, which affected daily activities or required prescription pain relief and a potential visit to a healthcare provider. This is not to be overlooked. The vaccine will ensure an uncomfortable few days for some. There will be a small subset of the population for whom COVID would have been asymptomatic, and the vaccine will prove a greater disruption to their lives than the disease it aims to prevent. It will be wise to present this information to clinicians and the public in a clear and direct manner. The safety of the vaccine outside of the periods immediately following an injection was promising. This table shows the number of unsolicited adverse events in the 28 days following either dose. When controlling for events related to the study and the vaccination, 8.2% of the vaccine group experienced an unsolicited event compared to 4.5% in the placebo group. The vaccine saw more events compared to placebo. However, as a proportion of the population given the vaccine, the figures are small. There were no fatalities associated with the vaccine, and a comparable number of serious events were noted in the two groups. We now come to a critical question. Can the results of this study be applied to the UK population? The study was conducted across 99 centres in the US, and the study population was recruited local to these centres. Using the data from the participants, we can see that the study population has much in common with the UK population. There is a rough similarity in terms of gender, age, ethnicity, and COVID risk. The stratification ensured that the key categories of 65 and above, under 65 and at risk, and under 65 with no risk were adequately represented so that valid conclusions could be drawn from each group. 42% of the study population were above 65 or at risk, and this stands up well against the 20.5 million patients on the NHS's shielded patient list for being at risk or high risk of COVID-19. There are a couple of caveats, however. In terms of occupation, there was a strong bias towards healthcare workers, with 25.1% of participants in the study being healthcare workers. And for this reason, the health literacy and health behaviours of the study population are unlikely to reflect that of the UK population. Secondly, the paper is clear in its exclusion of pregnant women and children, but on closer inspection, in trying to capture this, they have enforced a barrier curbing recruitment from the female, fertile, and not pregnant group. The commitment to contraception or abstinence for a period of 5 months during and around the study will have proved a deterrent for some of this group. The magnitude of this effect is impossible to interpret from the data, but unlikely to have a significant impact on the primary outcomes. However, it is worth noting as there is potential for improvement in the study design. The next question asks if all clinically important aspects were considered. The paper considered both efficacy and safety, with a number of secondary outcomes, most notably the efficacy against severe COVID. There are some relevant clinical outcomes that the study was not able to explore. First, the participants were followed up for a median period of 63 days only. This allows for little information on the long-term efficacy of the vaccine. The waning of efficacy has been demonstrated in other vaccines and is an important consideration from a clinical standpoint. Secondly, the nature of the study means that it offers hazy insight into the effect of the vaccine on asymptomatic COVID. Asymptomatic COVID is thought to account for a significant proportion of COVID infection, and there is evidence that asymptomatic individuals are the most likely to spread the virus. Given the efficacy shown in the study, it is intuitive to think that efficacy will cross over to asymptomatic individuals. However, this paper does not provide the evidence to assert that conclusively. SARS-CoV-2 has thrown the world into a crisis the like of which has never been seen. A safe and effective vaccine offers light at the end of the tunnel. This study presents a serious contender for a safe and effective vaccine with a particularly noteworthy short-term efficacy. There are some issues with the study. The unnecessary missteps with regard to blinding, a tone that doesn't seem interested in dwelling on the adverse events that will become some patients as a consequence of the vaccine, and limitations on the time frame that participants were followed up for, which leaves us unsure about the long-term efficacy of the vaccine. However, when viewed in the context of a global pandemic that has brought nations to a standstill, there is much to celebrate about this study, and I side with the general consensus that there is a clear advantage to approving the vaccine and incorporating it in a nationwide vaccination strategy. Vaccine efficacy must be differentiated from vaccine effectiveness. How effective the vaccine is in practice will depend on the implementation of a well-thought-through vaccination strategy that aims for high levels of vaccination in the population. The final paragraph of the paper makes a point worth ending on. Within 1 year after the emergence of a novel infection that caused a pandemic, a pathogen was determined, vaccine targets were identified, vaccine constructs were created, manufacturing to scale was developed, Phase 1 through Phase 3 testing was conducted, and data has been reported. This process demonstrates what is possible in the context of motivated collaboration among key sectors of society. Thank you very much for your time. I hope you enjoyed the presentation.